The overall goal of this procedure is to analyze leg muscle physiology, its timing and kinematics in a tethered insect during different walking situations. The stick insect, Carausius morosus, is used for this study because, for an insect, it is relatively large and has clearly separated segmental ganglia. First, the animal is tethered and labelled for fluorescent video tracking. Next, EMG electrodes are placed in the protractor and retractor muscles. Then, the preparation of the tethered insect for tarsal contact recording on a slippery surface follows. The final step of the procedure is the simultaneous recording of leg muscle activity and tarsal contact during different walking situations. Ultimately, results can be obtained that show alterations in muscle timing depending on the behavioural context a leg is used in.
The main advantage of this method over existing techniques like studying walking on trackballs is that mechanical coupling through the ground is removed.
This method can help answer key questions in motor control, such as how much a single leg contributes to walking in a multi-legged animal.
The implications of this technique open the field of adaptive locomotor behavior, because we can now study changes in walking speed and walking direction on the single leg level. Demonstrating the setup will be Hans-Peter Bollhagen, a technician in my laboratory.
Two firmly joined nickel-coated brass plates, insulated by each other through a 2 mm wide strip of 2-component epoxy, are used. To prepare the slippery walking surface for the animal, lubricate each half of the plates.